In this webinar series, this activity aims to expose all BS Biology program students to research areas in biology, biochemistry, and molecular biology. So this is to prepare you for your upcoming thesis subjects. The speaker in this webinar are researchers and professionals of different fields of science. So they may also come from international platform, but as of today will be our speakers from the local platform. So please listen to the speaker, and your teacher might require you a synthesis paper. So we will also have a question and answer part later. So you question the speaker about his presentation. So let me introduce you our speaker for today. So our speaker for today was cum laude when he graduated Bachelor of Secondary Education here at Nueva Vizcaya State University in 2010. He finished his first master's degree, Master of Education, Science Education, also from NVSU in 2016. Then just this year, he got his second master's degree, Master of Science in Chemistry, major in Medicinal and Biological Chemistry from National Chongqing University in Taichung City, Taiwan. During his stay at Taiwan, he published several research works concerning the cellular mechanisms of Protectobacterium carotivorum, subspecies carotivorum. He is expert in antimicrobials, bacteriocins, biopest control, cloning, gene knockouts, PCR, pharmacognostical analysis, phytochemical screening, protein expression mycotoxins, and natural products. His current objective is to generate functional natural products using indigenous plant materials here in our province for the benefit of the community. So without further ado, let us all give a round of applause to Sir Ruchi, Brian, James, Sir Senya, Lagitna. Sir Ruchi.
老师可以看出来，老师可以看出来，是早上好 ，Hello， 我在来瑞奇，我是学生，来我的中心大学，来从台湾，跟化学老师，我的 Nueva Vizcaya。 Hi, I am Mr. Ruchi Brian James S. Lagitnay, Lai Ruchi, a graduate of the Master of Science in Chemistry from National Chongqing University, Taichung, Taiwan, and a faculty of the College of Arts and Sciences Natural Science Department, teaching in chemistry. For today's seminar workshop, I will be presenting to you the Pectobacterium carotivorum subspecies Carotivorum bacteriacea, exploring its related genes, functions, and regulatory mechanisms. Sounds like too technical, right? But don't worry, I'll try my best to come up with a presentation that can be easily understood, especially for those who don't have a deep background in biochemistry and cellular biology. Before we'll continue with the presentation, just to give you a brief background, I'll be delivering you a simple tour on the laboratory that we had in the chemistry department of National Chongqing University, particularly the equipment and the laboratory that we have under the medicinal and biological chemistry group of the chemistry department. Sorry for the technicalities. Rewind. So moving on, the title of my presentation is Pectobacterium keratoborum, subspecies Keratoborum's bacteriocins, exploring its related genes, functions, and regulatory mechanism. So sounds too technical, but mind you, these are the different methodologies and techniques that we utilize here is considered as the simplest among all the biochemistry and molecular biology techniques. Okay. So in our laboratory, we have the assigned office where students or Shueshang in Chinese uh, can do their assignment or present their meeting.
Tapay mo na yung data.
Ha?
Para kay Wanjag.
Tatang?
Datang.
Sorry for some technicalities.
Bukan dia berdapat dengan itu. Pero
Tuloy.
Okay, so what is being presented on the screen is a picture of our or the student's office. Adjacent to our office is our laboratory. So in our laboratory, we usually isolate our bacteria. We are only working with one bacteria, that is the Pectobacterium keratoborum subspecies keratoborum. From the isolation, we extract the cellular DNA or the genomic DNA and try to process it, amplify, we try to isolate the genomic DNA and amplify it by a polymerase chain reaction to come up with the different studies about the gene functions and regulatory mechanism. Okay. Of course, we utilize the polymerase chain reaction and the gel electrophoresis to check whether you succeeded in amplifying your DNA. Okay. So for this presentation, I have this outline, a brief introduction about the Pectobacterium keratoborum subspecies keratoborum. I will also be discussing what is bacteria. And then I'll be presenting to you the comparison studies that we did and the bacteriocin regulation mechanism, which is my master's thesis. So it dwells with the discussion on the isolation to the homologous recombination, the in vivo studies, and the in vivo and the in vitro studies, and the arrival of the conclusion. So if you happen to go to the market and check the different agricultural crops, particularly vegetables, after some time, the vegetable that you bought in the market will have this macerations on the plant tissues, resulting to what you call wet and often foul-smelling plant organ. So this observation for plants and vegetables is what we call the soft rot disease. So soft rot disease is caused by the bacteria Pectobacterium keratoborum subspecies keratoborum. So this is a phytopathogen primarily secreted that degrades the pectin in the middle lamella and primary cell walls that causes the macerations on plant tissues, creating a wet and often foul-smelling plant organ. So the bacteria reach high concentrations on the xylem and cause necrosis on plants' vascular tissue. So using the scanning electron microscope, we were able to determine the morphological characteristic or morphological structure of the bacteria, thereby classifying these bacteria as rod-shaped gram-negative. So Pectobacterium keratoborum subspecies keratoborum, or PCC in short, was formerly known as Erwinia keratobora subspecies keratoborum. So this bacteria is phytopathogenic in nature, meaning they don't cause harm to animal kingdom animalia and other organisms, but only for plants. This type of bacteria is easy to characterize by DNA sequencing using the 16S DNA. And in the laboratory, we can easily culture it at 28 degrees to 30 degrees Celsius. This bacteria also can be easily isolated using a semi-selective medium, which is the modified Drygalski's medium. So in our laboratory, we took the Pectobacterium keratoborum subspecies keratoborum in Brassica rapa subspecies pekinensis, or in Filipino yung tinatawag natin na umbok or Chinese cabbage. Okay. So this bacteria is resulting to worldwide economic loss in agriculture. But despite being phytopathogenic in nature, a lot of studies reveal that this PCC is a potential source of antibiotic called bacteria. So what is this? So what is this bactericin? So when we say bactericin, these are bactericidal and extracellular toxins produced by bacteria. In the natural habitat, in order for each organism to survive, they develop this mechanism to fight for foreign bodies or other microorganisms. So this bacteria produces a proteinaceous compound that inhibits the growth of other microbes in order for them to survive, especially in a lacking of nutrient environment. So bactericids are a miscellaneous group of protein compounds that are being produced by diverse bacteria, which usually demonstrates antibacterial activity against species of closely related to the bacteriocynogenic strains. Okay, so few examples of bacteriocins are mesins, plantasarins, bovisins, and colicins and the like. So colicin is one of the well-known bacteriocin that is produced from Escherichia coli strain. So what about the mechanism of action of this bactericid? Since I told you a while back that it only destroys related strains, but not the producer strains itself, this protein is secreted ribosomically by the producer strain. So the secreted toxin or protein may be in the modified or unmodified form. So, from the structure of the protein, it will be attached to the receptor protein of the target cell and may cause DNAs or RNAs. So, thus killing the target bacteria. For the DNA's activity, the bacteriocin will target the DNA of the target bacteria, thereby causing inhibiting mRNA translation or an RNase that inhibits protein translation from the mRNA. Then thus
Napapansin mo
killing
ba?
the target. Aside from antimicrobial, there are a lot also of studies being published which gives us the applications of bactericin. So bactericin nowadays are also used for cancer therapy, gene delivery, drug delivery, antiviral like the anti-malarial drug, antioxidants for tissue engineering, and anti-inflammatory. Now, since my topic is about the bactericin by PCC. PCC is a gram-negative bacteria. So here are the different classifications of bactericin produced by gram-negative bacteria. They are subdivided into four. We have the colicins that are produced by E. coli with a molecular weight higher than 10 kilodalton. These colicins are subclassified into subclass 1, which are forms 4 in bacterial cell wall, the subclass 2 bactericin that degrades the nucleic acid. Aside from that, we also have the colicin-like bactericin. These are produced by bacteria other than Escherichia coli and with a molecular weight higher than 10 kilodalton. Also, they are subdivided into subclass 1 and subclass 2, which forms 4 in bacterial cell wall and degrading nucleic acid. The third classification is the microcells. They have a molecular weight lower than 10 kilodalton and with the subdivision of subclass 1 with approximately or more than 5 kilodalton in size and are considered post-translationally modified, and the subclass 2 with a range of 5 to 10 kilodalton of molecular weight and are considered as unmodified or minimally modified protein. The last, which I know is very much familiar to all, especially those who knows about antibiotics or bactericin, are the phage tail-like. So these phage tail-like molecules are highly similar to the phage tail structure of virus. Worldwide, PCC is known. They are known producers of bactericide. In Korea, they have found out that PCC can secrete the bactericide kerosene. In United Kingdom, they have published a paper that reported PCC to produce pectocin M1 and pectocin M2. While in Japan, they were able to publish a paper reporting that PCC can produce high molecular weight phage tail-like bactericide, which is named as carotoboricin. In our laboratory, we also reported PCC as a producer of multiple bactericide, meaning one bacteria can produce either carotoboricin or the plantasin or the kerosene of different kinds. Particularly, we have the kerosene S1, kerosene S2, and kerosene S3, which in the bactericide assay could inhibit greater zone of inhibition, as what you can see on the screen, and are considered as low molecular bactericide. At the same time, PCC can also secrete carotoboricin, which is considered as high molecular weight bactericide, having a phage tail-like structure. So in the bactericide assay, we can observe the secretion of carotoboricin by a minimal or small zone of inhibition. So in the let me present to you first the common flow to study bactericide. How do we get this antibiotic in order for us to utilize whether as a biocontrol agent for the soft disease or as medicine? So the general flow is we isolate the bactericide from the producing strain. So usually we get the bactericide from an infected plant, either a vegetable or a fruit. And then from the isolation, we test its antimicrobial activity. And then from the isolated or from the tested colony exhibiting antimicrobial activity, we try to get the DNA, try to purify, express the protein using different molecular techniques, and try to study the different genes related on it. And afterwards, using other techniques, we can also characterize which proteins do they produce, or if it's a novel bactericide, then we can furthermore study its regulatory mechanism. In our study about the kerosene, in the recently published paper, kerosene S3 from BMC Microbiology in year 2020, here is the sorry for the technicalities. Okay, so this is the workflow that we utilize in our identification of the kerosene S3 as a new bactericide produced by PCC. So we started with the preparation of the bacterial strains, the plasmids, media, and the growth conditions. So after isolating the bacteria, we did some bactericide activity assay, and then the reverse transcriptase polymerase chain reaction for RNA transcription, simple Western blotting for kerosene S1, and then the candidate for the kerosene S3 was isolated for its genomic DNA, and then we designed the primer for the nested polymerase chain reaction for hybridization, and then use restriction DNA library screening and Salter blotting. And afterwards, we ligated it and do the bactericide assay, DNA sequencing, and some computer analysis for sequencing of data, and then subcloning the isolated gene transformed again into a bacteria, and then we do some protein expressions and bactericide assay activity, after which we purified again the protein from it, and then tested for its activity. And lastly, we utilize mass spectroscopy for the determination of its molecular weight assay or molecular weight determination. Don't worry, this might sound too technical for you, but as I discuss one by one the different procedures, at least you have some in-depth information about it. Okay. So, moving on. So from the method that I projected from the previous slide, this is what you call a bactericide assay of PCC strain. So from the single colony that we have selected in a semi-selective media, we try to assay it with an indicator strain and determine which colony produces high molecular weight bactericide and low molecular weight bactericide. From there, we took one colony, which is our candidate for producing the low molecular weight bactericide, which exhibited greater or bigger zone of inhibition. Then, we assign a name for each colony. From that bactericide assay, we took single colony and we named this H-Reef Paleo PCC or H-Reef 86, Pectobacterium keratovorum subspecies keratovorum, which is a wild type strain. From that single colony, we isolated its genomic DNA. Then, using different restriction enzymes, we try to cut the different parts of the genomic DNA of this H-Reef 86 PCC. Then, from that, we transferred the genomic DNA in an agarose gel. We have the candidate base pair of 4.5 kilo base pair gene isolated from the restriction enzyme EGL2. This candidate genomic DNA fragment is then subcloned into a cloning vector PBR322. So, genomic DNA fragment of DNA cut by BGL2 and then subcloned into a cloning vector. This is a plasmid that can accommodate foreign DNA. So, we name this vector PBRS3. So, upon ligation, in using ligation reactions and transformation and Southern blotting, we try to hybridize this plasmid with labeled DNA group. From there, using different molecular techniques such as DNA sequencing, we found out that this candidate gene named tyrosine S3 contains two open reading frames. It has a 2,640 base pair open reading frame for the killer protein and a 270 base pair open reading frame for the immunity protein. Okay. So, from the cloned tyrosine S3 containing two open reading frames having the killer protein and the immunity protein, we hybridized that and tested for bactericin assay for the confirmation if the cloned tyrosine S3 gene possess bactericin activity. So, what you can see on the plate is a colony for the H-Reef paleo or the wild type strain. The second colony is the E-Link Leopa or the EA1068, which is an indicator strain. This is also Pectobacterium keratoborum species. And then the inserted by homologous recombination PBRS3 or the gene bactericin gene inserted into the EA1068, which do not possess any bactericin activity. So from there, you can observe that the EA1068 alone does not possess any bactericin activity. But when we inserted the tyrosine S3 gene into the EA1068, you can observe clear and bigger zone of inhibition. Meaning, this isolated gene that we have in our DNA library is a bactericin. Okay. So how do we know that? Okay. We have different models using different softwares that use amino acid sequencing and other platforms that use omics in biochemistry to determine protein structures and protein activity. So tyrosines are considered as CLBs or colicin-like bactericins. The second classification of bactericin produced by gram-negative bacteria. So, we have found out that tyrosine S3 shares structural homology to that of colicin B, which is a bactericin produced by Escherichia coli. So in the structure of the tyrosine S3, we modeled that the tyrosine S3 contains a translocation domain, a receptor binding domain, and a killer domain. So what is this? These are the parts of the bactericin that are necessary for the attack, translocation, and regulation mechanism. So in the receptor binding, this portion of the protein is responsible for at binding the target bacteria. And then the translocation is the one that is responsible for the flow, how this bacteria will get inserted into the target bacteria. And lastly, the killer domain will be the one responsible to attack or inhibits the growth of the bacteria. Now, in the killer domain, there are a lot of ways how bactericide kills. In our case, we have found out that our bactericide, carosine S1, carosine S2, and carosine S3 possess DNA structure. So, as a DNase protein, they inhibits the translation of mRNA. From there, the regular function of the target bacteria will no longer work because the DNA is already inhibited to translate into mRNA, thereby causing the lysis or the death of the target bacteria. So that's how our kerosene works. Okay. So we then investigated this killer domain of the kerosene S3. So from the purified kerosene S3 killer protein, or known as caro S3K, that is our coding for the killer protein. Using sodium dodecyl sulfate phage analysis, we transferred the expressed protein from Escherichia coli BL21 and then transferred into the SDS and utilize Malditos MS for the characterization. Malditos means matrix assisted laser desorption, ionization, time of flight, mass spectroscopy. So just to give you an idea how this Malditos work, we all know that biomolecules possess different molecular weights. Okay, so this polypeptide will then absorb energy from the system of the Malditos. Since these proteins possess different, the components of this polypeptide possess different molecular weights, this Malditos then will measure the time of flight, how each molecule will travel in the matrix of the Malditos. So, based on this, we can determine the exact molecular weight of the protein of interest or our target protein. Now, based on our Malditos data and with our SDS page, we have confirmed that the molecular weight that has been observed in the SDS coincides with what we have taken in the Malditos MS analysis, with a 95.6 kilodalton molecular weight. Same is with the immunity protein or the caro S3. The isolated purified carosine S3 immunity protein that was expressed in a cloning vector and an expressing bacteria E. coli BL21 strain has a molecular weight of approximately 10.2 kilodalton. Okay, so we have confirmed that this immunity protein has this molecular weight. Sorry, medyo shortcut itong ating presentation because we will be having too long in discussion if I am to give you the full detail of each step. So I don't expect you to grasp all, but at least just to show you how our research is being done, and if ever that you would like to make also such research later on, at least now you have this first glance of what is this research all about. And as you go along with your courses later on in your biology courses, you can be able to at least touch the principles may be involved in RT-PCR, PCR, gel electrophoresis, what is SDS page, how do we purify protein, what is this cloning and the like. So, but for now, maybe just a brief introduction on this kind of research. Okay, so we have confirmed the molecular weights of tyrosine S3 and killer protein and tyrosine S3 immunity protein. Now, aside from that, using the different in vitro and in vivo tests that we have come up with the mechanism how this bacteriocin works. Okay, so being a nuclease bacteriocin, the tyrosine S3I and the tyrosine S3K in the producer strains are both secreted outside the cell. Now, into the target cell, when this tyrosine S3 in complex, when we say in complex, tyrosine S3 containing the killer protein and the immunity protein. When this tyrosine S3 complex enters the cell, what will happen? Nothing will happen with the cell. Why? Because we have this immunity protein that inhibits the killer protein to attack the DNA of the cell. Okay. But if we isolate the immunity protein of the bacteriocin, this will cause now the DNA's activity. It will now harm the DNA, causing the target target bacteria rather to degrade its DNA, thereby causing lysis or cell death. Okay. Now, how this bacteriocin from PCC will be transported to the target bacteria. So PCC adopts what you call type 3 secretion system. So this type 3 secretion system is a unique secretion system of all the compounds or all materials that this cell produce. Say for instance, this toxic bacteriocin or toxic protein. Okay, so just to make the presentation simple again. Okay, so what you see on the screen are the different parts within the outside, the inner outside the membrane, within the inner membrane, and onto the hook of the bacteria, the producer bacteria. Remember a while back, I have provided you some image of the PCC. So it has a hook or just like a flagella. Okay. So because PCC adopts type 3 secretion system, we have two types of hook. We have the flagellar or the hook type. Okay. In our recent research publication, just this November 2021, we have reported that PCC has two types of secretion system, the type 3A and the type 3B, which is the hook and the filamentous secretion system. Okay, so the secreted bacteriocin will then be attached into a secretion signals and will be involved in some enrichment and sortings by a chaperone uh, RNA, and then it will be attached into the AP base complex for the cargo formation, and this will be secreted via a proton motive force driven injection. So meaning we don't need a lot of energy for this bacteriocin to be ejected out of the cell. It will just be natural because of the proton motive force due to the electron charge that this bacteriocin is produced. So these are the essential parts of the type 3 secretion systems that are that will be utilized in order for this bacteriocin to be secreted outside of the cell. Okay, so among the important parts are the FLHA, which will be using later on in the other part of the presentation. We have the FLHA, FLIT, and then we also have the serine, the AP base complex, and the RNA chaperone and the like. So these are essentials for the regulation mechanism. Okay, we have the fly-C, the fly-G. The fly-C is the gene responsible for the filament. The FLHA is responsible for as a gateway that will initiate the proton motive force. The fly-G for the attachment into the serine, and okay. So these are the essential things. Now, what is this BRG gene? Okay. We now proceed with how this bacteriocin is being delivered and how it is being regulated by the producer strain. So in my research work, I utilize this BRG gene. So what is this BRG gene? So this BRG gene is homologous to the host factor for bacteriophage or the Q beta RNA, or commonly known as the HFQ. So what is this? This is an RNA chaperone involved in a wide range of cellular process pathways. Okay. So this type of protein is pleiotropic in nature. So have you heard about the term pleiotropic? So when we say pleiotropic, pleiotropic genes are, or pleiotropic gene is a multi-function protein. Okay, so when we say pleiotropic, it's multi-function. So this BRG binds the small non-coding RNAs, or the sRNAs, and are therefore recognized as a transcriptional regulators of global gene expression. So they plays a pivotal role in the control of gene expression in bacteria, either positively or negatively. Okay. So thereby, this controls the translation. Okay. So the inactivation then of this HFQ gene, or the BRG gene in the PCC, has been found to have a broad pleiotropic genotypes affecting growth rate, cell morphology, and cell tolerance. Okay. In other researches, they utilize BRG to make a virulent strains or species. Okay. So when we say a virulent, what does it mean? So they don't possess already the characteristic of being harmful to other organisms. Take note, Lactobacterium keratoboreum subspecies keratoboreum is a phytopathogenic bacteria, right? So they infect bacteria and they induce soft rat disease. Yung tinatawag natin na what's that in Filipino? The soft rat disease, yung, what do you call that? Soft rat. Pagkabulok. Okay, yung nabubulok yung vegetable. That is their nature to attack the host, which is the vegetable, okay, of any kind. So binubulok nila either the root or the stem. Basta they are highly concentrated in the xylem and in the phloem of the plant. Okay, so if we try to create or to transform PCC into a virulent, so what will happen? If we try to put the PCC, the virulent PCC, into the vegetable will no longer have the soft rat disease. But instead, okay, it will no longer have, it will no longer cause the soft rat disease, but this virulent PCC will still attack other bacteria present into the vegetable. So we can utilize now this PCC as a biocontrol agent against soft rat disease. Okay, ganun yung ginagawa. This is the reason why we characterize the protein, the bacteriocin we produce. Why do we study the regulation mechanism on how PCC excretes or secretes the bacteria and how this antibiotic is being utilized within the cell. Okay, so one focus is for biocontrol. Another is we can utilize this for medicine, particularly human medicine. If we try to study the different gene related to the bacteriocin, then we can come up with a very good drug delivery system for better utilization. Okay, so moving on, in this study, we try to discover the effect of BRG at the transcriptional level. As the BRG gene, the cloned version of HFQ, we will now determine what will be the influence of BRG on the synthesis of the bacteriocin, the regulation mechanism of BRG towards PCC's bacteriocin production, and what is the function of this BRG in the type 3 secretion system. Okay. So with this is the methodology that I followed. Okay. So the BRG was already subcloned, and so they were isolated from the plasmid, and then I did some plasmid confirmation, and then to confirm whether the inserted gene is correct, I do sequencing, and then multiple alignment, and then do the homologous recombination, after which I did some recombinant screening, and then I used 2 induction for the expression of the protein, either by ultraviolet induction and the glucose induction, and then I did some in vitro and in vivo observations and then data analysis. Okay. let's proceed with the cloning of the BRG. Just like what I have told you a while back, the genomic DNA of the PCC was studied, and we have these DNA libraries. Okay? So since we already have this data, we try to isolate the BRG gene using restriction enzyme. Okay? We cut then the portion of the DNA and try to transform it into a cloning vector. This time, we utilize the PGMT-EC vector. So this is a plasmid that can attach or can accept foreign DNA structure or foreign DNA fragment via TA cloning technique. Okay, so from there, when we inserted the BRG, we now have the PG-BRG plasmid. From the PG-BRG plasmid, we try to knock out the BRG. Okay, so how do we do the knockout? A knockout is an experiment wherein, okay, I'll go back. Okay, if that is the full DNA sequence or fragment, full DNA fragment, what will you do in order for you to knock out this? We try to cut a big portion of this DNA fragment and insert an antibiotic cassette or of any kind. And so what will happen if you try to insert that at the middle of this DNA, it will inactivate the function of the protein. So that is what you call gene knockout. Okay. So that was the experiment that I did. So in this case, the PGBRG was inserted with a tetracycline drug cassette. Okay. So using the site-ligated method or site-directed mutagenesis, yung tinatawag natin na SLIM method. So we inserted the tetracycline. How did we do that? We cut the fragment of the BRD gene using the restriction enzyme ZO1 and HPA1, in which the ZO1 and HPA1 also is a restriction enzyme of the tetracycline cassette, so that you can easily use that into the PGBRG. So from PGBRG, naging PGBRGT, since it already contains the tetracycline cassette. Okay. So this PGBRGT is 4,796 base pairs in total. Okay. Okay. So here is the electrophoregram for the different processes that I do. So in the detection of the recombinant vector, in figure A, that is the 4,796 base pair BRG knockout cloned in the PGMT-EC vector. And then upon digestion with ZO1 and HPA1, we have separated the fragment into a 3,015 base pair, sorry. Excuse me for that. So, using the ZO1 and HPA1 restriction enzyme, we have separated the two fragments of the vector, the 3,015 base pair vector, and then the 1,781 base pair, which is the BRG gene. And then, using the nested PCR, we amplify this 1,781 base pair BRG gene. Okay. After the knockout, using restriction enzyme and polymerase chain reaction, this plasmid was reinserted into the PCC H389H12, which is the wild type strain that I utilized. So, using homologous recombination technique, this now plasmid will try to join via homologous recombination into the wild type DNA, into the wild type DNA. And so, from the homologous, as a result of the homologous recombination, the wild type strain can now have the tetracycline gene inserted into the original portion of the BRG gene. Okay, so what will happen? From there, we can now observe a big difference into the activity of the cell. Based on our screening of the BRG knockout, okay, so this is the homologous recombination screening of the mutant. So the first plate are the successfully transformed BRG mutants after electroporation. So we inserted the DNA fragment via electroporation. So we utilize electricity to drive the DNA fragment for the DNA fragment to get inside the cell. And then within the cell, there is this natural phenomenon of homologous recombination wherein the homologous DNA fragment will have switching or attachment to the parent DNA. Okay. So since the tetracycline gene is the one that we utilize, now this mutant will now exhibit tetracycline, or this mutant can already grow in a tetracycline rich medium. Take note, tetracycline is an antibiotic. So since this is an antibiotic, if this is just a wild type strain, basically this strain cannot grow into that medium. But if homologous recombination is successful, then this strain now can have this tetracycline gene that allows the bacteria to grow into a tetracycline rich medium. Okay, so that's letter B, the different single colonies that we have there. And then, so from colonies 1 to 4 are the wild type. So as you can see in the colonies 1 to 4, no growth of PCC was observed. But for the rest, from number 5 to number 40, colony 5 to colony 48, all of them grow. And then the negative control there is the Escherichia coli from colonies 49 to 52. So that is one way of screening the homologous recombination. Aside from that, to check whether or not this bacteria is really Pectobacterium keratoborum, we utilize the modified Drygalski's medium. Okay, because if this is just Escherichia coli or other contamination, then using this modified Drygalski's medium, we can say that there is a contamination within the process of the homologous recombination. So it's not the PCC. So in the modified Drygalski's medium, the color of the colony is lighter white, lighter blue to greenish color, while for Escherichia coli, it will be dark blue. Okay, so good thing, based on the plate, colonies 5 to 48 all pass with the screening. Aside from that, we also have a DNA sequencing using the 16S for the confirmation, for the last confirmation if this transformed mutant is correct, is PCC or not. Okay. Okay. So after the homologous recombination was screened, next is the in vivo study. Okay. So what do we have here now? So this is now the confirmation of the homologous recombination mutant, the delta BRG mutant, and that of the wild type. Based on the bacteriocin assay that we had, we can see, and under the influence of UV irradiation, the wild type possess, of course, possess the bacteriocin showing zone of inhibition. But for the mutant or the knockout gene, it no longer possesses zone of inhibition, which means that the secretion of bacteriocin is already inhibited. Okay. Same as true plus but a different observation was seen when this bacteria, both the wild type and the BRG mutant, was inoculated in a sugar-rich medium. Okay. Let's proceed with the transcriptional analysis. Okay, so as demonstrated in this transcriptional analysis of bacteriocin genes and the regulatory genes by two-step endpoint reverse transcriptase RT-PCR under the UV irradiation. Okay. We have observed that on the knockout strains, transcripts for tyrosine S2K, transcripts for tyrosine S3K is no longer visible. Okay, so this is now the reason why we say that our knockout is correct or is working, because it inhibits at the transcriptional level the transcription of the Caro S2K, which is the killer protein for the tyrosine S2, or the killer protein for the tyrosine S3. Moreover, the different regulatory genes, such as the fly-C, okay, is also not exhibited into the transcript of the transcriptional analysis that we did for the gene knockout. But if you are to check the transcript in the growth of or the transcript under the glucose induction, we can say that there is a big difference into the transcripts of all the necessary genes, Caro S2K, Caro S3K, Caro S4K, the CRP gene, the DGC gene, and the FLHA. These are the regulatory genes. The GLNH and the YSAT, these are the type 3 secretion genes. Okay, and the GLNH and the YSAT. So based on the transcripts, we can see that the trends in the transcripts are fluctuating under the different concentrations of glucose, meaning under a nutrient rich environment, the work of the BRG differs. But under the ultraviolet induction or UV exposure, the BRG works well. Okay, so why do we use UV induction and the glucose induction? Because basically the plants are exposed to sunlight. Sunlight contains UV. Okay, so of course, this UV from the sunlight can cause them the BRG to work in the environment strains. And then why do we have the glucose induction? Because of course, within the xylem and the phloem or within the cell this, of the plants, the producer bacteria are exposed with glucose. That is the reason why we tested the we mimic the environmental conditions of the bacteria within in the vegetables. So that's the reason why we utilize the glucose and the UV. And so from the transcriptional analysis, now the final, we have proposed with a mechanism how this BRG as a RNA chaperone gene. Okay, so this BRG gene does not influence the CRP, which is responsible for the production of the CAMP that activates the secretion and the regulation of the bacteriocin. This also BRG does not directly regulate the DGC, which is a component within the cell that is responsible for the synthesis of the CDGMP, which controls bacteriocin production. Okay. Now, being an RNA chaperone, this BRG is then utilized by the ATPase complex as a chaperone in the delivery. This one will convert or modify the structure of the kerosene, either kerosene S1, kerosene S2, or kerosene S3. It will modify its structure so as this kerosene could fit into the type 3 A secretion system, so that upon the modification, this protein can easily get inside the components of the type 3 secretion system and be ejected by a proton motive force outside the cell going to the target cell. So that is how BRG influence the secretion of the bacteriocin within the cell. In conclusion, we have found out that PCC is a multi-producer of the low molecular weight bacteriocin, since we have reported that the PCC strain can secrete kerosene S1, S2, S3, S4, and the metabolism. Bacteriocin kerosene S3 is born by chromosomal gene, which encodes the killer protein and an immunity protein. And by the SDS phage analysis, it revealed that the relative mass of kerosene S3K and kerosene S3I were 95.6 kilodalton and 10.2 kilodalton respectively. And the BRG knockout was unable to produce the low molecular weight bacteriocin, and the kerosene production of the BRG knockout mutant is inducible by ultraviolet irradiation, but not the addition of glucose. And so we have concluded that BRG is potentially required for kerosene externalization through the type 3 secretion system. Thank you very much. I would like to acknowledge Tranche Laboratory in National Chongqing University. They are my lab mates. Of course, my next please. My ever supportive lab mates, my ever supportive family. Sorry, lab mates, lab mates, and of course my support group. Okay. We have the National Chongqing University, Huaxiaxi, or the chemistry department, the Department of Science and Technology, because they were the one who granted me the scholarship to study abroad in collaboration with the MECA TECO, the Manila Economic and Cultural Office, Taipei Economic and Cultural Office, and of course, my ever beloved Nueva Vizcaya State University and my college, my sending institution, College of Arts and Sciences, particularly special mention to the Natural Sciences Department. Of course, my support group, members of the Church of Christ, the local congregation of Titan City, and lastly, the Taiwan scholars, Pinoy Taiwan scholars. So they are the Filipino scholars, postdoc, PhD, masters, and college students studying in National Church University. Thank you very much.